Today on Zenith, it's a condition that strikes hundreds of thousands of Americans, yet there is no cure. Can research about to begin not only create a way to study multiple sclerosis, but potentially offer ways to find a cure? We'll hear from a professor whose research is giving us new answers into this and other conditions. It's all next on Zenith.
This program has been made possible by a grant from the UCF Office of Research and Commercialization.
The UCF Office of Research and Commercialization is committed to moving the discoveries of our faculty and students from ideas to innovation to realization. By moving research from the laboratory to the private sector, we are helping to diversify Florida's economy and helping to bring high-paying jobs to our state. This program presents some examples of our research and our efforts to transition this research to the private sector.
Hello and welcome to Zenith, the program of and about research at the University of Central Florida. I'm your host, Ed Hyland. Today we will look at exciting discoveries into the material that coats our very nerves and hear about a research project that might unlock clues into understanding and treating conditions like multiple sclerosis. But first, before we look to the future, we'll take a look into the past and a way of seeing history that has been hidden from our eyes for centuries. Arlen and Diane Chase have been plowing through the wild tropical forests of Central America for over 25 years. Together, they've given us an understanding of one of the largest Maya cities. Caracol was lost to archaeologists for hundreds of years until the Chases began digging it out inch by inch. Slow work, a costly time investment, but now they are able to see this ancient wonder through new eyes, called LiDAR, light detection and ranging. The images this surface scanning technique can produce are stunningly detailed and gives new focus to the length and breadth of Caracol. Dr. John Weishempel helped develop and is analyzing the LiDAR data.
Pretty much everyone knows what a laser pointer is, right? So if you imagine that instead of a single beam of light, comprised of many, many photons, imagine many laser pointers kind of grouped together, shining light down. Okay. Now, a laser pointer is half the instrument. The other component is you're actually timing the speed of light. And so what that means is your laser pointer is now a rangefinder. It can measure distance. So you send down light and record how long it takes for it to reflect off an object and come back to your receiver. And so that gives you a range. And so now if you're sending lots of photons down, you're able to map ranges on a broad scale, and we know that as topography. So LiDAR is basically a remotely sensed instrument using lasers to measure topography or surfaces. I've worked with LiDARs in the past where the resolution was much coarser. We were able to, the resolution was on the order of 15 to 20 meters horizontally. The vertical resolution is still less than half a meter, very good. But now these data, the resolution's on the scale of meters, and that's the horizontal, and the vertical resolution's probably on the order of centimeters. And when I first got the data set, it was pretty funny because I got this JPEG that was sent to me, and I looked at it, and I thought, yeah, okay, no big deal. But then I started zooming in, and I'm zooming in and zooming in and zooming in. And all of a sudden you see buildings, causeways, terraces, and then every once in a while you'd see that little pivot or divot, and it's like, what is that? Then we went to the point cloud data, and we were able to really say, oh, that's a cave, or some kind of opening into the ground. But it really was amazing, the number of features that we could see, and the real kind of I want to say the clarity of it. I mean, it was very clear. Not being an archaeologist, I'm looking at this stuff and I'm saying, there's all kinds of stuff there. And then finally, when I took it to the archaeologists, they were definitely taken aback. Diane and Arlan have been working 25 years in this area, very intensively in about a tenth of the area that we've flown. But even in that intense area that they studied, we're showing them things they've never seen before. Because all of a sudden you have this top-down perspective, and things that on the ground it's hard to say, well, that's a square, But when you're looking from the top down, you say, oh yeah, that is a square, and then it brings out some features that they couldn't see. So the LiDAR is able to bring out features that may only be this high off the ground. So we're working in a tropical rainforest in Belize, fairly high, canopy cover. But if you go into a forest and lie down and look up, you'll see there's little openings in the canopy. That's why there are occasionally plants on the ground, because there's holes in the canopy allowing light to come through. And so all canopies allow certain degrees of light to come through. Some can be pretty dark, but they've measured canopies in the Pacific Northwest, and they're still able to penetrate all the way to the ground. So if you have enough energy, blast enough photons down through the canopy, there are holes there, and so eventually some of those photons will make their way down and reflect to the top. For every square meter, we sent down roughly 30 photons. Okay, only 1.5 photon actually would make its way on average to the ground, and so you do then interpolate in between some areas. Some areas where it's more dense, you may have less than 1 photon per square meter. Areas where it's less dense, you'll have more. And so based on the very last or lowest return, then you can interpolate across the landscape and then produce the ground topography, or they call it the bare earth topography. If you look at all the data, it's called the point cloud. I mean, in our area we did roughly 200 square kilometers, and we had like 4.5 billion shots. Okay, most of those shots are canopy. So I mean, I'm able to look at forests as well as look at the ground. So I can measure things like tree height. I can measure things like tree diversity or structural diversity of the trees. And so if you're a monkey, that may be useful information to you. But if you're an archaeologist, it's kind of noise. So you want to eliminate it. So this allows us to sample without destroying the forest. So we're able to really see through the trees. My work has been looking at forests, measuring trees and canopy structures using lasers, but this is the first time that we used it with archaeology. And it really does represent the most extensive use of lasers to look at below canopy archaeology. There's been little groups of people here and there looking at other archaeological features below what I'd probably say would be less challenging canopies. This is, I think, fascinating. We didn't expect this. Now the Maya, they looked at caves as portals to the underworld. They had this underworld idea, heaven, hell, not exactly, I'm sure I'm misinterpreting it, but the underworld was, below the ground. And caves served as ritual sites. And what we were able to do, we're coming across, looking at the topography, and all of a sudden there's a huge pit, down there. And some of these holes went as low as, or as deep as 80 meters. And so that meant that the airplane must have been, or at least the lasers, coming in fairly vertically. Now we certainly can't measure caves if it's a side opening, but if it's a vertical opening, we measured probably somewhere between 60 and 100 different openings. And these openings were at least 10 meters deep below the ground surface. And so we've only started exploring just a couple here and there. It's going to require some technical spelunking to really explore some of these caves. It's going to revolutionize archaeology. It really isn't that expensive when you think about it. So doing 200 square kilometers, the flights from Florida to Belize, the processing, it was about maybe 10 hours of actual flight time where you're measuring it, but the cost was $170,000. Well, let's compare that to 25 years worth of surveying in the jungle. There's a huge difference there. I mean, and how quickly you get the data is phenomenal. There is really very, I think, little limit in terms of price for that. I really see this just exploding in the next year. I mean, people are already calling. How can we do it? Can we do it?
LiDAR is generating interest not only from archaeologists, but also for its ability to map changes in ice caps and forest diversity and density. Yes, LiDAR could be a tool to measure climate change. A major NASA project has been funded with the goal of putting LiDAR into space by 2017. Coming up, for some medical conditions, there are drugs to treat the symptoms, but nothing to treat the root cause itself. One of those conditions is multiple sclerosis, or MS. But a team at the University of Central Florida is taking a closer look at the nanoparticle level into what causes MS and just maybe how to repair the damage it causes. That story is coming up on Zenith. A National Institutes of Health grant will help University of Central Florida researchers explore new ways to potentially reverse damage caused by multiple sclerosis and other neurological disorders. Stephen Lambert, an associate professor in the College of Medicine and a member of UCF's Hybrid Systems Laboratory, has received the first installment of a $1.9 million grant to study the breakdown of myelin. Myelin is a substance that coats and protects nerves inside the brain and spinal cord, enabling electrical signals to reach distant nerve cells and muscles. Without it, the brain signals are weak, confused, and eventually stop reaching critical cells. I asked Professor Lambert how the grant will be used to forward his research.
Well, it's one of the adaptations that higher animals and higher organisms have. So as the length of the axon, that's the bit that conducts the electric, it's like a wire. As that gets longer and longer and you build more complex nervous systems, you have to electrically coat that wire. And in order for it to actually transmit very fast, and in order not to have enormous heads the size of baseballs to accommodate all the wires that it would take to build that. So what we have is a series of cells that wrap themselves around that axon, around that wire, kind of like the plastic insulation that you would see on an electrical wire. And what they do is they electrically coat that, they support that cell, and then they allow for very rapid conduction through those wires. And that's essentially what myelin is. There's a separate type of myelin in the brain as there is in the peripheral system.
Now, do we know why it starts to break down or start to not work as effectively as it's supposed to?
Well, I mean, that's been one of the big questions in multiple sclerosis research for many, many years. There's sort of two fields, if you like. There's the immunologists, who believe that the answer really lies in the fact that your body starts to break down your myelin, you develop an immune response to the myelin. And then there's the sort of the neurobiologists on the other side, and you know, every 4 years we go to Italy, we get together, we drink a lot of wine and we argue about this. And those guys believe that there's essentially an initial trauma somewhere in the nervous system that then leads to an immune response and then the breakdown of the myelin, and then it becomes an immune disease. But everybody agrees on one thing, and that is that essentially the outcome is that it's an immune disorder. Your body starts to break down its own myelin.
And is that why it's been so difficult to treat? Everything we have right now seems to treat the symptoms.
That's exactly it. I mean, the way that the disease is currently treated is to basically try and stop its progression. And we try to do that by essentially offering a plethora of anti-inflammatory drugs and reagents against the immune response itself. And then, you know, that's a good idea because it's nice to be able to stop that as a progressive disease, but it doesn't reverse the damage that's already been done when one of those insults takes place, and that's essentially what we're trying to do.
And is it also somewhat different in each individual, so that complicates it even further?
The the pathology of how you manifest with MS, for instance, and I should say we're also trying to look at other central myelin disorders and even some peripheral myelin disorders as well, which I've been working on for 10 years. But the reason that the pathology is different in a lot of places, it depends where your lesion takes place. So for instance, if you have an assault essentially on the optic nerve, you're going to lose some vision. This sometimes may be temporary and it recovers, especially in the early phases of the disease. And then as you progress and you have more and more insults, this may not recover. A lot of people get weaknesses in one side versus the other. Some people have trouble talking. I mean, and again, during early phases of MS, at least, you know, there's going to be a recovery phase. You may have an insult, then you may recover partially or altogether from this. and then until you have your next insult, when it may actually hit you somewhere else altogether.
And do genetics play any role in this?
Well, there's certainly some data to say that that your genetics, in terms of how your immune response might take place, does have a role. There are certain what we call HLA types, which are mapped to the immune response. They're part of how the body recognizes itself, and those definitely have been shown to have some genetic disorders. But other than that, not really, no. And this is MS is the most common neurological disorder among young people. I mean, I'm constantly amazed by the number of people that come up to me and say, I have a daughter with MS, or I have a friend whose daughter has MS. I mean, it's a very, very common disease.
Now, what will the dollars that are flowing into the research program for you, how is that going to help further your research?
Well, so one of the things that currently is used to study the disease are obviously animal models for diseases like MS. And what we're trying to do as an overall goal in the Nanoscience Center is to essentially develop things in a culture dish that can be used to replace animal models. This will cut down on the number of animals that we use, and obviously it also strips away the complexity of working in an animal. And so what we're trying to do is actually build this whole complex phenomenon of myelination, how the cell wraps itself around this wire, this axon, and try to put that into a dish in a functional system so that we can actually test, well, what happens when you remove the myelin sheath, and how can you stimulate cells to go back and remyelinate and repair the damage that's been done by the disease. And for years we've been able to build these types of systems in the peripheral nervous system. Now I have with my partner Cristina Fernandez-Valle, we have grants on working on that out at Nona. But this is the first time but the central nervous system has eluded us. We find it very, very difficult to build these types of systems. It's much more complicated. And so this money will allow us to build these types of systems to essentially test new drugs, to develop hopefully work with people like at the Burnham to develop new drugs that will eventually lead to reversing the damage associated with MS and other central myelinating diseases.
Now, could you give us a time frame? And I realize everybody probably asks you that. When can you give us some time frames? But can you just kind of lay out how you progress with something like this? Because this is a multi-year grant that you're working with the National Institute of Health, and it's going to help obviously over a couple of years. But where do you start, and what do you hope? What are your benchmarks? Maybe it'd be a better way to put it.
Well, we have to. This is when we write one of these things. We obviously, we have those exact requirements. So where are you going to be at this time next year? Where are you going to be at this time next year? I mean, the first thing to get over is that this is going to be this is essentially a chip. That's how we like to refer it. We're trying to build a chip, and so on this chip there will be muscle, there will be neurons taken from the spinal cord, and then there will be the actual myelinating cells. And this whole thing will be patterned in such a way as that we're building a functional circuit that has a length of myelin in the middle. It has readouts. And Jay's lab has done some marvelous work on this already, and has actually
Dr. Hickman.
Dr. Hickman, sorry, yes, who's already actually built part of this circuit already, where we have muscle that we can measure the contractions on, almost like little diving boards built out of silicon. And so what we're doing now is the next phase of this will be to modify this and introduce the myelinating cells, get the whole thing working in terms of electrical readouts, both in and out. And what we'd like to do is essentially have at least the basic circuit laid down in the first 18 months. So I mean, at the moment, and it's kind of an unusual group because I'm a basic scientist, I'm a basic myelin biologist. Jay is a bioengineer. Some of the people that we work with in nanoscience are chemists, and so it's kind of a multifunctional approach. And sometimes we all don't speak the same language. So especially when you have a German engineer, he doesn't speak any. But at the moment, the idea is to basically go through and design these chips. We're still at that design phase now, altering what we currently have to fit the myelinating cells. And as I say, I think within 18 months, we'd hope to have a functional circuit. And I'd like to be at a position in probably about year 2 to 3 where we actually start in a position where we can start to test drugs that are currently may be used to see how we can reverse some of these myelin processes. So that would put us in the final year of basically getting some really nice results from this system.
Now, when you create, within this 18 months, you create this functioning circuit, what you hope to accomplish there, how do you compare that? I mean, how do you make sure it's an accurate model? What do you compare it to? Because as we've discussed in previous Zenith programs, the animal models don't work. So what do you compare it to?
Right. No, this is actually a very difficult. What we would use is we would use some hallmarks. I mean, one of the basic things is can we get myelination to take place? Does it look like functional myelin? Does it change the way the axon conducts? And those parameters are being built into the chip. So we will have functional readouts that say, yes, it works and not. And then the other thing is, when we demyelinate, can we reverse that procedure? And we will compare it, obviously, with what is currently used in the animal models. The problem with most of the animal models is, for instance, they have to be virally stimulated to achieve demyelination, and the demyelination is often so extreme that it's very difficult to actually reverse it in the animal models. Either that or they're genetic models, which obviously are not the cases with most multiple sclerosis. So the idea is to build a functional circuit, compare it at least in terms of its function with what happens in the animal, and then use that as our benchmark going forward.
Now, in trying to do some research on that, it comes to mind that there are other conditions that possibly could be myelin related. Are there spin-offs? Are there things that perhaps can come out of this work as well?
I mean, if you look at most of this press, I think it's talked about multiple sclerosis, but in fact, this system is designed not to just look at central, but also peripheral myelin. There are a number of what we call neuropathies that actually are affected both in the peripheral system. So, for instance, one of the side effects of diabetes is a peripheral neuropathy, which causes a demyelination. And I know Dr. Hickman's particularly interested in looking at it. He has a colleague with this disease. He has a passionate interest in looking at this disease. And so that's essentially another area that we're looking at, is also the peripheral. And I've worked with Christina for well over 10 or 12 years on looking at peripheral myelination. So I also have an interest in that area as well, and in looking at sort of things like polyneuropathies and stuff like that.
I'm thinking, I guess, Parkinson's possibly, muscular dystrophy, I believe, was another one that popped up.
That is actually, yes, so for the chip that's originally developed, you can actually use it to study not just myelinating. I mean, we're actually adapting the chip that Dr. Hickman's lab has been working on to look at myelin, and that was the basis of this grant. But the original chip can be used to study diseases like muscular dystrophy, and I know, again, that Dr. Hickman's got an interest in putting sort of dystrophic muscle on the chip and then looking at the functional effects of what happens in a dystrophic muscle. He has a beautiful system essentially for measuring the ability of the muscle to contract in a very, very simplified fashion. And so therefore it's got that basic circuit has a large number of applications that he's working on.
Now, past the 18-month stretch where you hope to be able to complete the circuit, so to speak, and you'd mentioned that you wanted to perhaps begin some drug testing at that point along the way. What's the process there? I mean, do the drug companies line up for something like this? Do you solicit people to come and create drugs specific to this model?
Well, I mean, you basically, the first thing to do is, I mean, obviously some of the drugs that are currently under development, or that are currently used will not apply to this model. I mean, we're particularly looking at compounds which we know will reverse the myelination defect and stimulate remyelination. So there will be only a limited number. Most of the drug companies obviously focus on controlling inflammation. That's where a lot of the interest in this has been, and we're not really looking at that. I mean, that's not something that we can actually model very well. And so what we'd like to do is model remyelination, which is, to me, actually is a crucial phase of controlling this disease, is repairing the damage. So what we would probably do is initiate, I mean, if there were compounds available that were available from drug companies, obviously we would welcome, some input there, and there may be such, drugs being developed. But we would also probably try to get together. In fact, we have Dr. Hickman has a number of collaborations. I do too with people at the Burnham who have these very large devices for high throughput screening and the generation of large numbers of combinatorial drugs based on what's available. And what we would probably do in a phase 2 is get together with the Burnham and people like that and try to test, to use ours as essentially as a high throughput system. In other words, we can test large numbers of drugs in a very, much more cheaply than you would if you had an animal model, and use that to try and identify and assess new drugs.
How valuable is that cooperation, that collaboration, and really that proximity to a Burnham, a College of Medicine?
It's essential everywhere. I mean, I came from the Northeast, where I was in the greater Boston area when I was out in Worcester. I mean, that being able to just get in your car and drive for 10 minutes and meet together with another group that are maybe approaching this problem from a different way, I think that's essential to research. I mean, research is a collaborative business. I mean, if you look at what we're doing in the Nanoscience Center, I mean, we have basic scientists, we have electrophysiologists, we have chemists, we have people who just computer model. I mean, and that's the kind of approach, I think, that if you're going to get on top of a complicated disease like MS, that's the type of approach that you need to take. So.
And does the recognition, there's been some pretty good coverage of your work and what's taking place and the grant coming out, does that kind of do a here we are kind of thing where you perhaps get some added attention that perhaps could generate some additional research dollars and maybe even some side work as well?
Well, most definitely. I mean, there's two aspects to this. One of the things that I always find is as a basic scientist, we sometimes forget that the endpoint is a patient. And what I get really moved by is that people write to you and they say, you know, I have a colleague or I have a daughter or something, and that to me is very important. That helps us focus on what the endpoint actually is, and you know, we can get scientists can get very easily sidetracked. You know, oh, that cell's doing something interesting, or that's doing something interesting. When you've got somebody writing in and saying, my child has had this for a number of years, I appreciate you guys getting on board with this, and it helps give you focus, and it helps you remember why you're actually in the lab and what you're supposed to be doing. So that bit's very nice. I really enjoy that, hearing from these people. And on the other side is essentially, yes, it's also nice if your colleagues suddenly say, oh, wait a minute, we've been working on this drug, and look, there's a bunch of guys down the road. And I think, especially for this area, as the medical city builds up and as Central Florida becomes much more high tech, I think that is going to be very important, these types of collaborations, as we get to talk to each other. I mean, for instance, Scripps down at Jupiter Beach has a beautiful high throughput system. Perhaps when they read the newspapers or whatever, they'll say, oh, we could be helping you with this. So those types of collaborations are very, very useful. I mean, a lot of what the Burnham has, for instance, is very expensive high-tech equipment that I could never afford. And so you have to have these collaborations in order to take the next step forward.
According to the National Multiple Sclerosis Society, about 2.5 million people worldwide suffer from MS, including about 400,000 Americans. Miss a Zenith program? Check out the archives online at www.ucf.tv and scroll down the program guide to Zenith. We'd also like to hear from you with questions or comments about this or any other program on UCF TV. My email is on our website, ehighland@mail.ucf.edu. The goal of research is to better understand the world around us. Our goal is to be a window to that world. For Zenith, I'm Ed Highland.